Hi, welcome to this week's lesson. All cells need energy, which typically comes from the breakdown of glucose via cellular respiration. Cellular respiration has three main phases: glycolysis, the Krebs cycle, and the electron transport chain, where glucose is converted into adenosine triphosphate, ATP, which cells require to do their jobs. It's kind of like going to a foreign country. Even though you have American money, you must convert it into a form that can be used in that country. Similarly, cells must take glucose and turn it into a form the cell can use. Sometimes the human body does not have enough glucose because of fasting or a low-carb diet. But wondrously, the human body is capable of making glucose from various precursors. In this lesson, we will explore the first step in cellular respiration, glycolysis. The somewhat reverse of glycolysis, we call that gluconeogenesis. The specific role of Aldolase and its isoforms and how they work in these two processes. And lastly, how is Aldolase gene regulated in these processes? Let's take a quick moment and one more time look at the objectives of this lesson. First, we're going to review the pathways of glycolysis and gluconeogenesis. And since they're so similar, we'll focus on all of the steps of glycolysis, and then we'll just point out how gluconeogenesis differs from that process. We're going to look at Aldolase's role in glycolysis and gluconeogenesis. By the way, Aldolase goes by several different names. We're going to compare and contrast those different isoforms. Where are they? What do they do? We're going to look at how they're expressed, and lastly, how is it regulated, specifically by several different transcription factors listed on that slide. Before we discuss how glucose is broken down or formed, let's discuss why glucose. Well, glucose is important to living organisms. Photosynthetic organisms make glucose for those organisms that cannot convert the sun's energy into the chemical compound. Across various life forms, glucose is used to make complex polysaccharides that are used in the extracellular matrix and cell walls. Glucose acts as an energy storage location when glucose is converted into glycogen, starch, and sucrose. We can take glucose and completely oxidize it to make ATP, which all cells use for biochemical reactions. We can also use glucose in the nucleic acid and NADPH synthesis when the pentose phosphate pathway makes ribose 5-phosphate. Because glucose is vital for life, organisms either make it via photosynthesis or obtain it from their food sources. What if an organism cannot make glucose and they don't get it via the diet? Well, gluconeogenesis provides a pathway. Why should we care about glycolysis and gluconeogenesis? Well, let's take a look at both processes and why they're important to life. Now, first, glycolysis means sugar splitting. It's the process by where a 6-carbon glucose molecule is broken into two 3-carbon pyruvate molecules. Now, pyruvate stay in the presence of oxygen is to be broken down into carbon dioxide. And all of the electrons from glucose are transferred to high-energy electron carriers like NADH and FADH2, which take the electrons to the electron transport chain where ATP is produced. Now, ATP, you recall, is required for coupled biochemical reactions. Glycolysis is a very exergonic reaction. It releases energy. And even when there is no oxygen, glycolysis still provides a small amount of ATP via fermentation. Now, some organs rely upon glucose, but it's not always available. While stored glycogen can be converted into glucose, sometimes that storage is inadequate. So the body uses gluconeogenesis to form new sugars from precursors such as lactate, pyruvate, and glycerol. In humans, this mostly occurs in the liver. Now, when there is excess glucose, glycogen is formed and stored. However, these storages can run out when the diet is lacking in carbohydrates or after fasting or exercising. In fact, after intense exercise, lactate produced via fermentation in the skeletal muscle goes to the liver where it's converted into glucose and then moved back into the muscles and stored as glycogen. This is called the Cori cycle. So in other words, sometimes the body needs to create glucose, and it can do that. Before we get into the nitty-gritty of glycolysis, let's take a look at some of the highlights of this pathway. Now, glycolysis occurs in the cytoplasm. And again, it's the breakdown of a 6-carbon glucose into 2 pyruvate molecules, both of which have 3 carbons. The 10 reactions of glycolysis are split evenly among 2 phases. 5 reactions make up the preparatory phase and 5 make up the payoff phase. Now, the preparatory phase requires 2 ATPs to begin converting glucose, so they're used. But it's okay because the energy content of those intermediate molecules results in the release of energy in the second phase. That second phase, again, is called the payoff phase. Well, not only are 2 pyruvates made, but 4 ATP molecules are made in this part of glycolysis. Now, recall we used 2 ATP. We make 4 ATP, so we get a result of net 2 ATP. Now, that's not nearly as exciting as the entire cellular respiration process, which can make 30 to 32 ATPs. Perhaps more importantly are the 2 NADH molecules. Now, these are those high energy electron carriers. They are made in glycolysis. Each molecule can carry 2 electrons to the electron transport chain, where the electrons power the proton pumps, which create a gradient, which then leads to the functioning of the ATP synthase molecule, which produces ATP. The whole process is tightly regulated via ATP consumption, NADH creation, the regulation of several enzymes, including hexokinase, PFK1, pyruvate kinase. It's further controlled by hormones like glucagon, epinephrine, and insulin. And again, changes in gene expression can also control the process. Let's go ahead and take that detailed look at glycolysis. Now, we start with glucose being converted into glucose 6-phosphate by hexokinase. This is the first step that requires ATP. From here, glucose 6-phosphate is converted into fructose 6-phosphate by phosphoglucose isomerase. This next step, this third reaction, also requires energy from an ATP. And you can see that is where the 2 required ATPs come in. In this step, fructose 6-phosphate is converted into fructose 1,6-bisphosphate by phosphofructokinase, or PFK1. As the name kinase implies, this enzyme adds a phosphoryl group from ATP to the molecule. In the fourth step, aldolase, which goes by a variety of names, including fructose 1,6-bisphosphate aldolase, converts the fructose 1,6-bisphosphate into 2 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now, this reaction with aldolase is called an aldocleavage and is reversible, and it will play an important role in gluconeogenesis. We'll also examine aldolase in greater detail later in the presentation. Now, there's one problem here in glycolysis, and that is that the glyceraldehyde 3-phosphate is the only one that can continue on with glycolysis. So this is where the 5th step in the preparatory phase comes in. Triose phosphate isomerase converts the dihydroxyacetone phosphate into a glyceraldehyde 3-phosphate. Thus, this part of glycolysis ends with 2 glyceraldehyde 3-phosphates that can then go through the payoff phase. Now, those glyceraldehyde 3-phosphate molecules are converted into 1,3-biphosphoglycerate by the enzyme glyceraldehyde 3P dehydrogenase. Aren't all these big names fun? Notice this is where 2 NADH molecules, those high energy electron carriers, are produced. And they're going to go on to carry 4 electrons to the electron transport chain, where they power the proton pumps and make all that ATP. Now from here, phosphoglycerate kinase converts 2 ADPs to ATPs. So if you recall, we already used 2 ATP molecules. So the generation of these 2 ATP molecules leaves us at a breaking even point of view. So we've used 2, we've made 2. Then the 3 phosphoglycerates are turned into 2 phosphoglycerates by phosphoglycerate mutase. Then Aldolase comes around and converts this into phosphoenolpyruvate. And finally, pyruvate kinase converts the intermediate into pyruvate and converts 2 more ADPs into 2 ATPs. Thus, the net amount of ATP is 2. From here, pyruvate can either enter the Krebs cycle in the case of fermentation, it can just go on into fermentation and become lactic acid or lactate. There are 3 main regulatory steps in this pathway, and they are controlled by steps 1, 3, and 10, which are controlled by hexokinase, phosphofructokinase, and pyruvate kinase. So again, glycolysis is split into 2 phases. You have the preparatory phase and the payoff phase. We use 2 ATP molecules. We make 4 for a net of 2. And typically, this is followed up by the Krebs cycle, where those 3 carbon molecules are further broken down into carbon dioxide. All of the electrons are harvested on NADH and FADH2, and they go to the electron transport chain, where ATP is made in much greater quantities. Before we look at the details of gluconeogenesis, let's just look at some of the highlights of this pathway. Now, gluconeogenesis is how the body can take pyruvate or acetate or lactate or some other molecules and actually create glucose. This mainly occurs in the liver, and it is an expensive process because energy is put in to ensure that the process is irreversible, not to mention you're building a molecule. In fact, this requires 4 ATP molecules, 2 GTPs, 2 NADHs, all to take 2 pyruvates and make 1 glucose. But it provides glucose for the brain, muscles, and red blood cells. The body has this unique ability to make exactly what it needs. It does not make glucose in excess. Even though 7 of the 10 reactions and enzymes are identical to glycolysis, there are 3 reactions that are significantly different. So you cannot say that these 2 processes are simply reverses of themselves. They are not. In glycolysis, there are 3 irreversible steps, steps 1, 3, and 10, as they're very exergonic. Therefore, gluconeogenesis needs to bypass these 3 steps. Now, these 2 processes do not occur simultaneously. If glycolysis and gluconeogenesis occurred at the same time, the overall reaction would be ATP plus water makes ADP, inorganic phosphate, and heat. No metabolic work would be done under that scenario. Therefore, the timing of these reactions are tightly controlled. Let's go ahead and take that deep dive into gluconeogenesis. Now, I know I said glycolysis and gluconeogenesis are not reverses of each other, and they're not, but 7 of the 10 reactions are. So let's just focus our attention on these 3 reactions highlighted in yellow, because that's where the difference occurs. Now, in gluconeogenesis, we're going to start at the bottom. And in this case, we're going to take 2 pyruvate molecules and turn them into phosphoenol pyruvate molecules. We can abbreviate that PEP. Now, this step is actually more of a series of steps, and I want to go into that in a little bit further detail here in a minute. Now, just know that this is what happens when you start with pyruvate. If you were to start with lactate to create glucose, you'd have a different series of steps here. So let's take a further look at how pyruvate is turned into PEP. Pyruvate is moved into the mitochondria from the cytoplasm, where pyruvate carboxylase and coenzyme biotin turn it into 2 molecules of oxaloacetate. However, this molecule cannot leave the mitochondria. So malate dehydrogenase uses NADH to convert oxaloacetate into L-malate, which can leave the mitochondria through a transporter. And then in the cytosol, it is reoxidized to oxaloacetate. And then PEP carboxykinase turns it into the PEP molecule. The reoxidation process produces an NADH molecule, but the creation of PEP requires a GTP molecule. So that is actually that very bottom step. Pyruvate does get turned into PEP, but by a slightly different process than is shown in the diagram. Now, the second step that differs from glycolysis is when we convert fructose 1,6-bisphosphate to fructose 6-phosphate. This reaction is catalyzed by fructose 1,6-bisphosphatase, or FBAase1, or fructose bisphosphate aldolase. There's so many different names for this enzyme. We're just going to call it aldolase. Now, instead of a phosphoryl group transferred to an ADP, a phosphate is hydrolyzed from the molecule and an inorganic phosphate is created. Finally, the last step that differs from glycolysis is when glucose 6-phosphate is converted to glucose by glucose 6-phosphatase. This does not require ATP since the phosphate ester is simply hydrolyzed and it releases an inorganic phosphate. And this is what makes it an exergonic reaction. I mentioned earlier in the presentation that we were going to take a deeper look into aldolase and its role in both glycolysis and gluconeogenesis. So let's just take a brief overview of where we see that in both processes. In glycolysis, fructose 1,6-bisphosphate is turned into two molecules by aldolase, and that is the glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate. Now, don't forget that the glyceraldehyde molecule is the only one that can go on through glycolysis. So triose phosphate isomerase comes in and turns the dihydroxyacetone phosphate into another glyceraldehyde molecule, and the process continues. Now, in gluconeogenesis, we're going backwards. So one of the glyceraldehyde 3-phosphates is first converted to dihydroxyacetone phosphate before aldolase converts it back into fructose 1,6-bisphosphate. Aldolase also has a role in fructose metabolism in the liver. So we're going to take a little bit closer look at just what is aldolase, all of its different acronyms and different names. How is it expressed? Where is it expressed? And how is it regulated? There are several different isoforms of aldolase. There are also 3 classes of aldolase. Class 1 occurs in plants and animals, and we can further subdivide this class into aldolase A, B, and C. Class 1A exists in archaea, class 2 exists in protozoans, fungi, yeast, and algae. So obviously we're going to focus on class 1, aldolases A, B, and C. But just for some background information, class 1 and 2 differ in their structure and in how they function. Class 1 uses lysine at the active site to create a Schiff base intermediate with a substrate. These form homotetramers. But class 2, on the other hand, well, they're metal dependent, typically zinc ions or other 2+ ions, and they stabilize the intermediate when the substrate binds to the active site. These form homodimers, and they're much smaller aldolases than found in class 1. But in general, aldolases have many functions, both enzymatic and non-enzymatic. We will focus mostly on the enzyme abilities of aldolase in glycolysis and gluconeogenesis. But it is interesting to note that they do play roles in cell scaffolding, signaling, transcription, and motility. Let's look at these isoforms of aldolase. Aldolase A is created by the aldoa gene on chromosome 16. It is expressed in the red blood cells and muscles. Aldolase B is transcribed from the aldab gene on chromosome 9. Interesting. 4 of these aldolase B enzymes have to bind together as a tetramer in order for it to function. Aldolase B is expressed in the liver, kidney, and small intestines. It is found in the liver where gluconeogenesis occurs, and to a lesser extent in the kidneys. Aldolase B is also found in the liver because this is where fructose metabolism occurs, and B is capable of breaking down fructose. A cannot. There is now some research that suggests aldolase C might be able to, but for the most part, aldolase B is responsible for the breaking down of fructose. Aldolase C is made from the aldoc gene on chromosome 17, and it is expressed in the brain, smooth muscle, and neuronal tissue. C seems to have a somewhat elusive role, but might play a part in cancer cells. So we saw there are 3 isoforms of aldolase. I thought it'd be nice to kind of take a look at how they are similar, because aldolases play a significant role in both glycolysis and gluconeogenesis. Well, it turns out all 3 have similar DNA sequences. As you can see, aldolase A and B are 66% similar, while B and C are 68% similar, and A and C are 78% similar. All three isoforms are also similar in their active site, size, and mechanism. They all have roles in glycolysis and gluconeogenesis because they all act on fructose 1,6-bisphosphate. But aldolase B, according to quite a bit of the research, has a greater role in gluconeogenesis. B also has a role in fructose metabolism. Some research suggests that aldolase C may break down fructose as well, especially when aldolase B is not found in sufficient quantities. We've already mentioned earlier that glycolysis and gluconeogenesis are tightly regulated. If they were to occur at the same time, there would be no benefit. Metabolically, no work could be done. They would kind of cancel each other out. So the processes are regulated through fast, quick, and reversible actions caused by allosteric effects, phosphorylation, and regulatory binding. They're also controlled by changing the amount of enzyme molecules through transcription factor. Now, we've already talked about the isoforms of aldolase, and aldolase B is a significant isoform because it's used mainly in the liver in gluconeogenesis and in fructose metabolism. It is also used in glycolysis as necessary, but its main functions are gluconeogenesis and fructose metabolism. Now, the ability to make glucose as the body requires it, especially in times of fasting or low carbohydrate intake, can be controlled by diet and hormones. Glucose and insulin increase its activity, and this allows sugar to be broken down to keep the blood sugar levels regulated, while glucagon and CAMP can down-regulate it, and this allows the blood sugar levels not to get too low. Well, what happens in the liver when one consumes a high carbohydrate diet? Glucose is broken down and used for energy, but the excess glucose is converted into glycogen or fat for storage. When short-term fasting, like while you're sleeping, glucagon will stimulate the conversion of glycogen into glucose to be used for fuel. Glucagon will also reduce glucose consumption, and this again helps to stabilize the blood sugar levels. But in terms of a longer fast, the body will have depleted its glycogen storage. So gluconeogenesis is required to supply the body with the necessary energy. The body can use fat stores and convert it into glucose as necessary. Aldolase B is required in the liver for gluconeogenesis as well as fructose metabolism. Thus, controlling glycolysis and gluconeogenesis can be managed by controlling the enzymes in those processes, in particular, aldolase B. So B can be further regulated through transcription factors such as HNF1, C, EBP, DBP, and HNF3. We'll go ahead and take a closer look at how those transcription factors work on the next slide. Before we look at how aldolase B is regulated by transcription factors, let's just review briefly what transcription is. Recall that RNA polymerase and transcription factors bind at the promoter, which is a sequence of nucleotides that indicate the beginning of a gene. There's also a region called the promoter box enhancer where the bases CCAAT appear. Now, there are different proteins or transcription factors that can bind to these regions. One is called HNF1. That stands for hepatocyte nuclear factor 1. Another transcription factor is the CEBP, where C references that cat promoter box, and then the rest of that stands for enhancer binding protein. There is also a transcription factor called DBP, and that is albumin D element binding protein. It's very similar to the CEBP. We'll talk about that in just a moment. There's also an HNF3 or hepatocyte nuclear factor 3, and they all work together to regulate the expression of aldolase B. Now, both CEBP and DBP belong to a class of enzymes with a basic leucine zipper domain at the C-terminus end. They are used in dimerization and DNA bonding. They both can bind to the promoter or enhancer regions of genes, and they're important in glucose synthesis. Now, the whole configuration where RNA polymerase and the transcription factors bind to the promoter is called the transcription initiation complex. Interestingly, aldolase B has a 200 base pair promoter region where transcription factors HNF1 and CEBP can bind. We're also going to see later that HNF3 can also compete and bind at that site as well. There is also a 400 base pair enhancer region where the HNF1 can bind. Well, in the liver, there's a cooperation they have shown through research between CEBP or possibly DBP being bound at the promoter and at least one HNF1 bound to the enhancer. So when you have one of these binding proteins on the promoter and one of the hepatocyte nuclear factors bound to the enhancer, this enhances the overall production of aldolase B. Research has shown that both HNF1 and HNF3 can bind to the promoter on overlapping sites. In other words, these two transcription factors compete with each other. HNF3 is a competitive inhibitor since it competes for the same binding site, basically, in the liver, and HNF3 binds more tightly to the promoter than HNF1. But when there is sufficient amounts of HNF1, it can outcompete HNF3. So this allows HNF1 to stimulate the promoter and allows for DBP-dependent transactivation to create more aldolase B. So HNF3 is an inhibitor. It can also inhibit the DBP protein, but non-competitively because they have different binding sites. Therefore, hepatocyte nuclear factor 1, DBP, CEBP are all transactivators which stimulate the production of aldolase B, while HNF3 can inhibit the production of aldolase B. So we have learned a tremendous amount during this presentation. We've looked at glycolysis and gluconeogenesis in details. We've looked at aldolase's role in both glycolysis and gluconeogenesis and mentioned that it also has a role in breaking down fructose. We looked at the 3 different isoforms of aldolase, where they're found and how they're expressed. We also looked at how aldolase is regulated so that glycolysis and gluconeogenesis are regulated, because again, we don't want them happening at the same time. We also don't want our blood sugar to go too high. We don't want our blood sugar to go too low. So regulating different enzymes, including aldolase by transcription factors, is vitally important because once again, cells require ATP, cells require energy, and so sometimes we need to break the sugar down to get that energy, and sometimes we have to go backwards and we need to build that glucose molecule for organs that might need it. But either way, the regulation of glycolysis and gluconeogenesis is super important to life.